Our joint and muscle receptors are critical to good movement through the sensory motor process of motor control. Muscles have two types of sensory receptors that assist us. One is located in the tendon of the muscle, and one is located within the muscle fibers. These sensory receptors are helpful to us for motor control when moving, but can also be helpful to use to assist therapy, as will be discussed in this presentation. Listed here are the major questions you should be able to answer after viewing this presentation. These questions will again be displayed at the end of the unit for your review. Pause the presentation in order to take time to carefully read and understand these questions. This is a drawing I made and took my time so that it looks certainly better than what I usually draw on the whiteboard in class. To the left is a hemisection of the spinal cord with major components identified. As the dorsal white matter is separated into the fasciculus gracilis and fasciculus cuneatus, we know this happens to be a cross-section of the spinal cord in the cervical region, as it contains the fasciculus cuneatus. On the far right side of the diagram, I've drawn some of the major muscle fibers responsible for contraction and relaxation of the muscle, and these muscle fibers are referred to as extrafusal muscle fibers. At the top of this portion of the graphic is the tendon, and you can also see I've drawn extrafusal muscle fibers which have been cut away. Within the tendon can be seen one of the muscle receptors, which is formally called the neurotendinous spindle, but is commonly called the Golgi tendon organ. Below this area can be seen what is identified as the neuromuscular spindle, commonly simply referred to as the muscle spindle. There are two types of small muscle fibers found within the fusiform shape of the muscle spindle, and therefore these are referred to as intrafusal muscle fibers, and thus the main skeletal muscle fibers are called extrafusal muscle fibers. There are two types of intrafusal muscle fibers, which are named by the arrangement of the nuclei at the central portion of the fibers. Therefore, one is called a nuclear bag intrafusal muscle fiber, and the other is called a nuclear chain intrafusal muscle fiber. In a classroom lecture, I would now hand out this page of the diagram without the labeling, so we can work together in drawing in the neurons which are part of the muscle receptor system, which connect the Golgi tendon organ and muscle spindle to the central nervous system and influence contraction or movement in the muscles, as well as send messages to higher centers for perception of muscle movement to assist with motor control. Beginning with the next slide, I will draw these connections for you. This diagram shows how the Golgi tendon organ serves as a muscle receptor. The receptor is found within the muscle tendon. As extrafusal muscle fibers contract and the muscle shortens, it creates a stretch on the tendon. The Golgi tendon organ is very sensitive, and as the tendon is stretched, it becomes thinner and presses on the Golgi tendon organ, which causes it to send a sensory response to the central nervous system. The neuronal fiber is referred to as a 1B sensory neuron and is excitatory. I've shown the cell body of the neuron being located outside the central nervous system. The neuron goes into the spinal cord at the dorsal horn. As shown, it goes into the fasciculus cuneatus to send sensory information up to the cerebellum and also the cerebral cortex to assist with motor control. It also connects with another neuron in the dorsal horn, which is an inhibitory neuron that influences the cell body of the lower motor neuron in the ventral horn. This is the alpha lower motor neuron, which excites extrafusal muscle fibers to contract. So when the muscle contracts, it causes a response of the Golgi tendon organ to send messages to higher centers and assist with motor control. It also importantly connects with an inhibitory neuron to cause the lower motor neuron to the muscle to not contract, which is why we have to continually send excitatory signals from higher centers in order to maintain a contraction, or the muscle will automatically relax, like dropping something that was in your hand if you fall asleep. The Golgi tendon organ can then be used to help influence excessive tone to help relax the muscle. This graphic shows a common splint used for the wrist and hand following a stroke which affects the upper extremity. Commonly, the extremity will have spasticity in the wrist and finger flexors. By wearing a hard splint that will help hold the wrist in extension and not allow the hand to close, will cause a reaction by the Golgi tendon organs of these muscles to send messages to the muscles to relax. October of 1982 was an important month for me as I began my research writing career with a simple case study article where I used plaster casting to make a splint to relax elbow flexor spasticity in a stroke patient. It turned out that I was the first one to develop this plaster casting technique, and it jump-started my research efforts. I've included the full article as an item you can download and read in this week's lesson. Though I'm a bit embarrassed by the simplicity of my research effort, it was an important beginning point for me in 1982. The other type of muscle receptor is the muscle spindle found within the muscle itself and is outlined in yellow in this graphic. As noted previously, it has two types of muscle fibers within it, the nuclear bag fiber and the nuclear chain fiber. The nuclear bag fiber responds to quick stretch on the muscle, while the nuclear chain fiber responds to submaximal resistance. Submaximal resistance, for example, is lifting a 1 kilogram weight as we are able to lift it. Maximal resistance would be trying to lift a 500 kilogram weight as we would be unable to lift it. Maximal resistance creates major output from the Golgi tendon organ, which will decrease muscle tone, whereas submaximal resistance will encourage contraction and strengthening. Quick stretch to a muscle, such as tapping on the muscle or vibrating the muscle, will activate the nuclear bag. This will help to increase contraction. Both of the intrafusal muscle fibers can therefore be seen influencing the cell body of the lower motor neuron to be excited and cause contraction. At the same time, these signals are also being sent into the fasciculus cuneatus pathway to send signals up to the cerebellum and cerebral cortex to assist with motor control and coordination. In the top graphic, the deep tendon reflex is shown as the reflex hammer is used to tap on the patellar ligament. This causes a quick stretch to the tendon and therefore to the quadriceps group, causing a quick stretch and resultant quick contraction of this muscle group. This could be helpful in therapy to encourage a weak muscle to contract by tapping on the muscle or using a vibrator to encourage the muscle to contract. This is due to the reaction of the nuclear bag intrafusal muscle fiber reaction to quick stretch of the muscle. In the lower graphic, we can see a patient strengthening her quadriceps on a weight machine, which is offering submaximal resistance. This will help to strengthen the quadriceps group, which is encouraged by the reaction of the nuclear chain intrafusal muscle fiber reaction. In this graphic, I am trying to represent someone being given a small, light package. Often when trying to decide what is in the package, we want to get a sense for how heavy it is. With a light object, we will often lift our hand up and down to try and get a good sense of the weight of the object. Why do we do this? It is obviously a submaximal weight that we are able to hold. So if we keep our arm and hand steady, we are getting feedback to higher centers from the nuclear chain intrafusal muscle fibers to help us understand the amount of resistance and thus the weight of the object. If we also move our hand quickly up and down, we are now adding additional information from the nuclear bag intrafusal muscle fibers due to quick stretch, which gives us an additional sensory feedback and more information to understand the weight of the object. As we know, the extrafusal muscle fibers are excited by the alpha lower motor neurons. As the intrafusal muscle fibers are also contractile fibers, they too have excitation by smaller gamma lower motor neurons. This is important as these muscle fibers must always have a slight stretch to the nuclei, so they will be able to react to any change in length of the muscle as a whole. If the muscle is lengthened, then these intrafusal muscle fibers are automatically stretched to give feedback to the central nervous system. However, if the extrafusal fibers are shortened, then the stretch is taken off the intrafusal fibers, and the gamma lower motor neurons need to excite the intrafusal fibers so they are able to again react to change in muscle length. This happens due to both excitatory and inhibitory neurons influencing both the intrafusal and extrafusal muscle fibers from higher centers. It's a balancing act to maintain normal tone. It is due to problems with these higher center influences that therefore creates both hypertonicity or spasticity, as well as hypotonicity or low tone. This will be discussed in greater detail in next week's lecture on spasticity. In lecture, I often refer to the gamma lower motor neuron system as the idling mechanism of the muscle. It's much like a car at a red light. When you stop at the light, the engine, or muscle of the car, must slow down or relax, but needs to maintain a level of idling tone to be able to react well when the light turns green. If the intrafusal muscle fibers are facilitated too highly, then the car is idling too fast, and when you take your foot off the brake when the light turns green, it will jump forward or contract too quickly. However, if the car is not idling high enough when the light turns green, it will take off too slowly or be hypotonic and also be a problem. Therefore, the gamma lower motor neuron system is very important to a good running motor or skeletal muscle system. This is a summary of the responsibilities of the different neurons referred to in this lecture. Pause the video and take time to read and understand the functions of each neuron related to the lecture. As at the beginning of the presentation, listed here are the major questions you should be able to answer after viewing this presentation. Pause the presentation in order to take time to carefully read and understand these questions.